morning, good morning. Yeah, so if I'm a little out of breath, I've just run from King's Cross. Travelling by rail in this country has always been fun. Ever since I've been doing a PhD in Glasgow, not a PhD, actually my undergraduate, and travelling around, it's always been a little interesting having to swap trains mid-station sort of thing. But yeah, today was another classic signal failure at Potter's Bar. But I'm here and I'm going to try and take you through, this works, through complement in atypical aHUS, and I'm going to start at the very beginning. I'm going to do some of the middle and then discuss whether it's the end of complement in aHUS as we know it. Now, when I was originally asked, and thank you to the organising committee for giving me the task of covering complement and aHUS, I was originally given the title of the known, the unknowns, and the unknown unknowns. And that reminded me, of course, of this famous quote, or infamous quote, by Donald Rumsfeld here, who was the 13th and 21st Secretary of Defense for the US. I'm not going to read it out. I'm sure you all know most of it. But as a scientist, we deal with these things all the time. There are things that we think we know. I think that's a better way to put it. There are certainly things we know are unknown. And of course, the unknown unknowns, well, they're everywhere. So you've got to love it. So how old is complement? Anyone out there want to guess? Anyone? What about if I put up this lovely dinosaur? As old as dinosaurs? It's actually twice as old as dinosaurs, so it's 500 million years old. So it's been around a very long time. It's an important part of our defense against the world, against bacteria, against everything else. It's one of the most formidable defenses, and it started back in this little critter, which is the horseshoe crab. Still around these days, but there are fossils that date back nearly 500 million years. And there's a small protein in there that's C3-like. I'm going to talk about C3 a lot. That's complement protein 3. As scientists don't have a lot of imagination. So number 3 on the cascade that I'm about to show you. So has much happened since that in 500 million years of evolution? Well, yeah, we've got 50 odd proteins now in the complement system. There are 8 which are involved primarily in recognition. So this is touch, sense, grabbing a hold of things, pattern recognition. There are, what happened there? Don't do that, eh? So there are 9 that are scissors that cut things up, molecular scissors. There are 7 that are building blocks, essentially, that just stick together and make things. Then we've got 11 proteins here that are interested in seeing what's going on, and they capture, they're receptors for all of these proteins that are built up and then are broken down by our scissors here. But by far the biggest group are our brakes. These are 2 different types of brakes. We have up to 18 types of brakes on the complement system, things that slow it down, things that keep it running at the right speed. The complement system, and here's the scientific version of it. This is one I use in my grants. Don't get worried, I'm not going to be asking questions on that. But this complement system is complex. There's that C3 molecule right in the center, the pivot of the system. It starts up at the top in these sort of pathways. This is an antibody structure here, so that's one of our recognition molecules, which triggers the classical pathway, and it comes down through here. And then we have, I've got ahead of myself by clicking that. Then we have MBL pathway, or a lectin pathway. That recognizes dying cells, essentially debris. So it's quite useful for cleaning up all the mess after an effect. And then we have the alternative pathway over here, which is this Clathrin wheel of really quick. Once that gets going, it could amplify really fastly. And that results then in things downstream, such as these flames here. So this is the heat, the inflammation, if you like. And these molecules are important for telling the immune system, the rest of our immune system, to come and get involved in a complement activation. And then there's another thing where we have lots of building blocks that come together, and this makes a pore in the cell membrane. And so if you have a holy cell membrane, it basically lyses, and you can destroy things like that using that. So our brakes come in here, all over the cascade, and they shut things off. And so you end up with this system sitting there quietly, ticking away. It does move, it goes slowly. You've got your brakes on. These things don't stop it completely. They're not there to stop the complement cascade. It ticks there, ready to activate as soon as anything invades. That's what the complement system's for. This is an example. Here's my expensive graphics all the way from Hollywood. Honest. So bacteria. So we've cut ourselves shaving, yeah? Most of you at some point in your lives will probably shave. And unfortunately, sometimes you'll cut yourself. And that opens up, gets through our first line of defense in terms of an immune system, which is our skin. There are other things that are defenses. These are barrier defenses, as we call them. So simple things, they're actually our first line of defense. But after that, it's complement. Complement is the next line of defense against things like bacteria. How does it work? So we're going to be talking about nasty bacteria, not the stuff we buy from Yakult or whatever that's good bacteria. These are the ones that are trying to do some harm. So we're in the blood. You've got to imagine that. Yeah, here we are in the blood. We've got our bacterial cell membrane, and this is our active C3. And it breaks down naturally, as I said. It ticks along and can bind to the cell membrane here. This fragment here, as I said, activates our white blood cells, so they get a bit angry. So they're ready to now go to action against the bacteria. We have various things happening that you don't really need to know, except all these proteins now build up on top of each other, one by one, slowly coming together. Well, actually quite fast. I shouldn't say slowly. It's slowly on this graphic. It happens fairly fast, in milliseconds. And we get a pore which allows water to enter the bacteria. Now, of course, it's not just one, it's many. So we have this amplification, rapid amplification, bang, there goes the bacteria. So that's what it's there for. That's what it does. And most of us, all the time, perfectly happy, no problem. It's kept in balance. So I think when we come now to the kidney, what we have to remember, of course, is that a cell membrane is a cell membrane is a cell membrane. It's been that way since the dawn of evolution. We've tweaked it as we've evolved. Obviously, there are different proteins in here. But in people, compared to, say, small animals or small organisms, but they're essentially the same. They're this lipid bilayer with things sticking out of them that are covered in carbohydrates, sugar, that are proteins. So they're essentially the same. And the kidney, by its very design, is absolutely packed full of blood and therefore packed full of complement. So if we didn't have our brakes, what would happen? So now we've got this particularly sensitive area, the glomerular basement membrane in the kidney, and we would have rapid amplification of the complement system, activation of our immune system, and destruction of the kidney, just like that. It would happen really, really quickly. So our regulators, of which factor H is one of the major ones, are so important for holding the complement system in check. Factor H is an interesting molecule. It's fluid phase. It's really at high concentration. It's one of the highest concentration proteins in serum. As well as being a fluid phase regulator, it has this area here for cell surface binding, so pattern recognition, so it's got a recognition, but it's also got the brake on the other end, so it's quite an interesting molecule in that respect, that it both binds specifically and can regulate the complement system. We have membrane bound, so these are tethered to all our cells, or some less or more, depending, and can go into a whole long story about which cells have them and which don't and why that's important. But for today, they're membrane tethered, they're everywhere. And this one, CD46, I think most people call it. This is its kind of old complement name that I still use. And then we have a molecular scissor which works in conjunction with these guys. It's called factor I. This is a a molecule that helps to break down C3 and hold everything in check. So let's run this again, and let's now put in our complement regulators. So factor H is good at coming along and just breaking up this C3 active molecule, so it stops it from going any further. It also is pretty good at binding onto the glomerular basement membrane here and protecting and stopping that C3 from getting any more activated. MCP can do the same job if it's on a cell surface. And factor I will then come in and break it all up and get rid of any of the C3, and we've stopped. So that is how the complement system is supposed to work, and will be working in most of us. But of course, I think I'm blasting through this at the moment. I'm still running to catch a train. So, of course, things don't always work out like that. And now we're coming to more what happens in atypical aHUS. So we have factor H, and this is our recognition area here. So I've crossed it out because we've got a mutation or a variant. And this really is how we became to understand that complement was really involved in atypical aHUS. It was finding a family with a variant at that particular part of factor H. And by the work that was done by Tim and people in Newcastle, we were able to establish that factor H was involved, and then we've now went on and established that how critical complement is to aHUS. So we have a variant in factor H. Now it can still go and bind in the fluid phase because it's still got that recognition. To some extent, it can still bind to C3 on the surface, but not as well. So we get lots more C3b binding, and therefore we get some activation of the complement system. Our brakes are now faded. They're not working nearly as well as they should. So the complement system is hyperactive. It's moving that bit quicker than it should be. It's more sensitive and will respond quicker. And we're also activating our white blood cells, as well as many other types of cell in the body are also, because of all of this extra chemicals, if you like, that are going into the system that shouldn't be, then the system is getting overloaded and becoming activated. And then nature, being the way that it is, has also provided one small chink in the armour. So I've said about MCP and I've said about surface tethering, but there is, at the glomerular basement membrane, it's not really a cell surface. It's more like plastic, a kind of plastic, a soft plastic or a flexible plastic. It's a layer that helps to support everything else. And it's entirely dependent on that fluid phase factor H. So if we have a weakness in factor H, there's nothing else to really step in at that particular surface. And that's really important. So this is just showing, this is just some staining of glomerulus, it's just showing factor H binding to the glomerular, this is staining for the glomerular basement membrane, and that's them together. You can probably just about make out the yellow staining, which shows that they're right on top of each other. So that's complement. In a nutshell, we understand, I hope we've got there in some sense, that complement is important for protecting that surface. But what else can we learn from our shaving incident? So let's go back to my high quality graphics. Bacteria get in. So they're now in the blood, and of course there are other things in the blood other than complement. We've got lots of these immune cells. I've already introduced my angry Pac-Man. So that's those cells there. We've got red blood cells. They play a minor role in disease setting, and I'll touch on that later maybe. And we have platelets. Now platelets are quite important in the sense that they are the things that help to generate blood clot. So this is the thing that stops us bleeding to death. The complement system and the coagulation system are linked. And so there are elements of the complement system activating and the coagulation system activating that integrate to help form these clots. Of course, clots are really important for our survival, because if we cut ourselves, if we don't clot, then of course we bleed to death. So clotting is another really important normal function. But when we come back to the kidney, then I'm going to try and bring it all together now in this one Hollywood extravaganza. When we come back to the kidney, now we've got this situation where we've got crosstalk between the systems, and a failed factor H just sails on past. See, it's gone. Didn't bind to the glomerular basement membrane where it's meant to, because it hasn't got that surface recognition. So it's not binding to the glomerular basement membrane. These are, so that's that area. We need to protect this area here. There's nothing to protect it, and potentially not so much on this side as well, on the side where the urine goes. We've got the blood on this side, and in our kidney, in the tissue. And so we're getting complement activation. Bang, 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 bang. Well, okay, it seems to be out the way. It's a little bit isolated. What's the problem? It's kicking off a lot of those chemicals, which in turn activate the cells. And what we're activating these endothelial cells. So that now creates a very rough layer which interacts with the platelets. And we get this clotting reaction now happening inside our body, inside our kidneys. The clot is also known as a thrombus. So we have this thrombus forming here. And again, the thrombus can interact and activate cells. And the endothelial cells activate cells. And you see what's happening here. We're just becoming more and more activated. Now, the immune system is designed to be activated and then to bring itself back into control. When that's done normally, that's not a problem. But if that continues to happen, what's happened is fibrin gets put down. So if we go from that flexible plastic, we end up with the kind of plastic, I was going to say the plastic in the chairs, but they're not plastic chairs. But you know what I mean, those really hard plastic chairs. It goes really hard and solid, and you wipe out all the structures of the kidney in the process, so the kidney fails. We know complement's involved in this because of the work that we've done with factor H, but we're not completely sure how you get from that kind of complement to this kind of effect, then back, and so on. So that's one of those known unknowns, in a sense. I'm going to switch gears now. Do you recognize who this is? Anyone? Yeah? So Will Self is, I don't know what he is, a writer, I think, and a comedian, a commentator. And I was looking on, as all people who give presentations, how do I express self? What is self? And of course this face kept popping up, and I thought, actually that is a nice happy self. So this here represents me, us, self, happy self. And I'm going to talk now what happens in immunity normally. So that's called tolerance. That white cell has just passed through normal happy us. So it's flying around quite happily. It's been educated. It's an educated white cell. It knows not to interact with us. It's just looking for those bacteria to come along. But of course, if we have our unhappy self here, then that can activate the white blood cells, or the white blood cells can interact with unhappy self to clean it up, to get rid of it. It sees that potentially as bad bacteria or something similar. This, of course, creates a lot of dead cells and potentially a lot of antibodies bind to this new dead stuff that's there, this new looking self. And that activates the complement system, and so that can turn the white blood cells against normal happy self. And in a nutshell, I hope to have given you autoimmunity. And that is autoimmunity in a simple fashion, that we get turned against self. Now, I've been working in Newcastle, as David said, on autoantibodies. So I've introduced antibodies in a kind of hand-wavy fashion. Antibodies are the things that we protect ourselves from things that we're vaccinated against, things that we've been immunized against. The reason we get immunized is to raise antibodies. Those are the things that float around the blood and bind specifically, we hope, to the pathogens. But what we found when you look in aHUS patients, and there was a group in France, I have to give them their dues, they were the first to discover this in 3 young aHUS patients. They found autoantibodies that Come on. There we go. Autoantibodies that bind to this area where we find most of the mutations, as I said, this recognition area. So the autoantibodies are essentially mimicking mutations. So mutations, variants are genetic. You inherit them. Antibodies are something that grow up within your lifetime. They're acquired. So this has happened through experiences that you've undergone. So whether that is some infection that you fought off, or whether that is something to do with the way that your kidney has been damaged, we don't quite know. But it's really interesting that these autoantibodies are generated and can, or are sufficient at least, to block the function of factor H. I don't know if we've definitively proven that if you have autoantibodies it will drive disease. And I'm of the mind that we're not completely sure whether the autoantibodies give you the disease or the disease give you the autoantibodies. So this is one of the great known unknowns that I'm trying to deal with. So we found these in 10% of our aHUS patients in Newcastle, and that number is replicated across other cohorts of people with the disease around the world. So we know that we have a loss of control of the complement system. We've known that for how long now? It's getting on for 15 years or so. More than that, in fact. And we as a complement community have been trying to devise ways of controlling the complement system. And you will probably be mostly aware of eculizumab. So I'm just going to run through how that works in our system, in my model system that I've developed. So we've got our variant here, and it comes in and blocks C3, and it's starting to take off. But then all of a sudden, we've got our antibody here. This antibody is one that we've injected into us. This is our eculizumab or Soliris. This is it binding here now and blocking the step to the membrane attack complex. So you don't get any of the holes. You still get a bit of activation, but now you've increased your brakes. You've brought your brakes back up, and you're bringing the whole thing under control, which allows the body a chance to recover. And that, in a nutshell, is how this drug works. But we are a little worried that we're still going to generate activated cells, and we're still going to deposit a lot of that C3b on the surface. It doesn't stop that, it just stops it progressing to making that membrane poor. So this is another diagram of the complement system. By now you'll be fairly familiar with it, and you could do this instead of me. Yeah? These are all the drugs that were getting developed, and this is 5 years old, that I've borrowed the slide from. So these are all the drugs that were out there that people are trying to use to target the complement system. So there is a lot of money, a lot of investment going in. People want to stop things at the C3 level. There are some pluses and minuses with that, because it is important for our immune system to have C3 functioning. So we've got to be careful how we do that. One of the things that I'm involved in is a molecule called mini factor H, which of course is what it says on the tin. It's a small version where I've taken the two functional ends and stuck them together. And that seems to work really well. It's just working well in the lab though, and it's nowhere near going to people. And a lot of these aren't. You'll see eculizumab or anti-C5 here, so it targets this part and stops going down to, I've lost the MAC, there it is, on there, the membrane attack complex, so that big pore. But if we can target with using maybe recombinant human factor H, putting properly working factor H back, or mini factor H, which I hope will take off because then my name goes up in lights at some point, that will help to bring that under control and stop from activating the immune system as well. Stop all that chemical getting released and triggering all our white blood cells to get angry at self. We don't want angry self, we want happy self. So I think in the near future we're going to be able to supplement the drugs that we currently have, and eculizumab is brilliant and has made a big breakthrough, but and then for that it is going to go down in history in the complement field for its for the strides it's made and the doors that it now opens for other complement therapeutics. Oops. So there is one fly in the ointment. So that was me just bringing the complement system to an end and say we don't need to worry about complement in aHUS now because we have eculizumab. It's maybe not that simple. This is taken from work done in Newcastle, and I know Tim's going to go on to speak about it in a bit more detail, so I'm not going to dwell on it other than to say this is our complement system. These are defects in genes, so this is inherited defects in our complement genes, so factor H is MCP, factor I and C3. And it makes up around 50-55% of the known causes of aHUS in those patients. But then there's this rather nagging unknown. So that's our definite unknown. We don't know what that is. We don't know exactly what they are. They may interact back through these genes. So it may actually be about something that alters complement. We just don't know. I could stand up here all day, and I've got a bit of time, and talk about the potentials. There's an interesting gene here that I don't really know a lot of because it's not complement and it's not immunity as I understand it. And then there's some novel genes, which I'll leave Tim to expand on. But one thing that has come out of this and is this gene that I said I don't know a lot about. It's reported to be interacting with this thing, the coagulation pathway. And as I've hinted and said throughout the talk, the coagulation pathway interacts with the complement pathway. And as a complement person for nearly 25 years, I'm ashamed that I've never really put this slide up before where I show coagulation as an activator of complement, as one of the activation pathways in the complement system. Because it clearly is. There's been a lot of work that shows that this coagulation pathway can interact and trigger, particularly through C3 and C5, and get the complement system going. So I'll just go on and just show you some of that data and some of that interaction just to bamboozle you before I leave you. So there's a little bit of evidence that it starts the classical pathway. There's a lot of evidence from all of these parts of our coagulation pathway that it can target C3 and cleave it, and once it cleaves it, once it gets that going, then of course the complement system will barrel on down and go to making MAC. So if we can't control that, then of course we'll get a disease. Equally, we can target C5. Now, one thing that I don't know and I couldn't find out while I was looking and I was really wondering is whether this bypasses eculizumab. That's a question that I will go away from here and go to the complement conference and keep that right at the top of my mind and see if that answer is out there, because I'm really intrigued to see if there's any evidence that this system would be able to bypass the function of eculizumab to block the deposition of MAC. It works two ways as well. So we can also, there's definite evidence that right at the top of this lectin pathway and right at the end of the complement system, the RIT can go back into the coagulation pathway and activate here and drive clot formation. So there's a lot of crosstalk between these two pathways, and there are a lot of genes and a lot of proteins, which means that if there's any defect in these proteins, just in the same way as there's defects in those proteins, then that can contribute to each other and lead to disease. And this is me at my summary, and I'm going to try and just take you back through and summarize that. So we've got our defective breaks, which leaves us with hyperactive complement. I think it's David who's going to discuss a bit more about triggers. Someone is. So these are the kind of triggers that can input into that and stress our system, make our body unhappy. We get uncontrolled complement activation, tissue damage, clot formation. That then will result in tissue damage and clot formation, and tissue damage and clot formation, and uncontrolled complement activation. So we're back to my favorite symbol, our Clathrin wheel, which I loved when I was growing up. This Clathrin wheel that is expanding and going beyond what our brakes are able, the remaining brakes are able to slow down, resulting in the onset of atypical aHUS. I think, from my perspective, and I would say, well, we're always told to say basic science. I would say I was a hardcore scientist and against the clinical scientists. That's my understanding, and thanks for listening. I'm sorry I went a little fast. I think the adrenaline from getting here was in my system a little. But I'm happy to stand here and take as many questions as you want to throw at me, and I'll try and answer them.
Thanks very much, Kevin, for a very concise talk and covering a very complex area. If anybody's got any questions, feel free to put their hand up. We'll run around with a microphone. If anybody has any of these forms they want to hand in, if they don't want to stand up, I'm quite happy to read them out and have them answered. The other thing all the speakers have said to me is, if you're too shy to come and stand up and ask a question, feel free to grab us during lunch. We're more than happy to chat about any specific questions you may have.
I get to run away, great.
Well,
yeah, sorry.
How many different types of aHUS is there?
How many different types?
Yeah, is it all one, or is it one and then there's like different things of it? I don't understand different strains. I'm not too sure on it.
I guess the simple answer is it's atypical aHUS, but of course there could be the familial form in the sense that you've inherited it, and then it could be an acquired form. So you've inherited it from your family, that a set of genes have been passed down to you from your family and have come together in a form that allows you, unfortunately, to get aHUS. Or it could be an acquired effect, and that would be something that's happened during your lifetime that specifically alters your immune system such that it breaks and you get atypical aHUS in that fashion. So I think there's broadly two types, I guess.
Yeah, I suppose what we think of as atypical aHUS here is what we're talking about is the complement-mediated form. But in actual fact, the term atypical aHUS is just a catch-all for anything that is not the typical form, which you'll understand is the Shiga toxin-associated form. So atypical aHUS is a broad church which encompasses many things. Some of these involve complement mutations, some of them don't. It could be just a drug-induced phenomenon. It could be pneumococcal, and not everything will have a complement-mediated cause underlying it. So atypical aHUS, as we probably understand it here, is complement-mediated, but in the larger medical community, it encompasses a lot of things which may not be. And we're happy to talk about that if you want to come up and speak to me later about the different forms.
Yeah.
When you talk about it's obviously a physical disease, but what about these sort of things that may cause it, the stress issues? Because the immune system is tied. There are ties out there between, for example, psychological stress, emotional stress, which can be severe, and then getting right into the actual physicality of disease. I just wondered if
I'm fascinated by that aspect, I have to admit. So hormone levels, changes in hormone levels affect the way that our white cells function. If you exercise a lot, if you don't, that changes the way our immune system works. When you age, that changes the way that our immune system works. I always think of chaos theory and think of the butterfly effect when I'm trying to explain how it all works, that tiny little things add up to a tidal wave somewhere. And I think that is what is going on. So when you're stressed, then it leads to lots of releases of things called prostaglandins and things like that, so chemicals. So that's that unhappy self again. There are all sorts of these chemicals, and they don't just act on one thing. They act broadly across tissues and different cells. Different cells respond differently, and say, something in the liver changes. That affects the secretion of factor H. So you get less factor H in your blood. So if you have less factor H in your blood, that's the same as not having the brakes on as much as you need. And that's a fairly simplified version of it. But if you think about all of the events that go on, I mean, flu is a good example, you know, because your body's under a huge attack. There's lots of cells dying. There's lots of cells being mopped up by the complement system, which essentially is soaking up factor H out of the system. So you'll get almost a local deletion or suppression of factor H because it's getting sucked away to do other things. So in that scenario, again, it's that dip which just allows the complement system to take off. So I think, yeah, it is really complex, and I don't think, I mean, I'm definitely hand-waving here about it, but that's my kind of feeling of how it all comes together. It is fascinating how the complement system and the immune system that sits sort of on top of it and above it, how they interact under stress conditions and under, whether that's emotional or otherwise. Yep.
You gave a very simple example of a cut shaving. This is a very naive question. But say somebody with aHUS has a cut from shaving,
Yeah.
what is the risk? Or is it just the damage to the kidney that causes it to kick in? I think that question makes sense.
Yeah, it does, and I hadn't pre-thought how to deal with that.
I mean, is there a risk of it kicking the immune system to such a degree that it will be necessary?
I don't think so. I think your immune system is got lots of safeguards and fallbacks. If the bacteria get in, which they've got a lot of hills to climb to get in for and establish themselves in the first place. There's a lot of barriers to go past. If they got in, and if you got an infection, then you're back into that group of people who, once they've had an infection, that can precipitate atypical aHUS. So, in the main, if you cut yourself, you'll be fine, 99.9% of the time. But 99.9.